Hello! Today we're going to be learning about gene regulation in bacterial cells, also known as prokaryotes. In order to get a better understanding of the mechanisms that occur within the cell, we're going to be focusing specifically on two operons, the tryptophan (TRP) operon and the lactose (LAC) operon. Before we dive too deep into the gene regulation involved with these operons, let's first cover the basic composition of an operon and its overall function. In a bacterial cell, clusters of functionally related genes can be controlled by an on-off switch, which can determine whether or not those genes undergo transcription and later on translation. This on-off switch is a segment of DNA called an operator, which is usually positioned within the promoter, which you might recognize as the place where RNA polymerase binds. Depending on whether the operator is switched on or off, it is either going to allow the RNA polymerase to continue to bind and carry out transcription, or it's going to prevent it by shutting down transcription of the gene. The operon itself is the entire stretch of DNA that includes the operator, the promoter, and the genes that they control. For the sake of this video, we're going to label this operon as the TRP operon, which includes the following genes, each of which encode the three enzymes responsible for tryptophan synthesis. By default, the TRP operon is on, which means that there is no tryptophan present in the cell's environment, which in turn causes the transcription of the genes required for tryptophan synthesis. However, the operon can be switched off by a protein called a repressor, which is the product of a separate regulatory gene, known in this case as TRPR. The repressor is able to bind to the operator and prevent gene transcription from happening by blocking the attachment of RNA polymerase. The repressor can be in an active, repressing transcription from happening, or inactive, allowing transcription to happen, form, depending on the presence of other molecules. However, the repressor is only active in the presence of its co-repressor, which is a molecule that cooperates with the repressor protein to switch the operon off. In this case, the co-repressor is the tryptophan itself. When tryptophan is present, it binds to the TRP repressor, which turns the operon off because it does not need to make the enzymes that will synthesize more tryptophan. When tryptophan is absent, there is no co-repressor to bind to the TRP repressor, and it is inactive, thus allowing transcription and tryptophan production to occur. Before moving on to the mechanisms of the LAC operon, we need to understand the difference between positive and negative gene regulation. The TRP operon is an example of negative gene regulation because it is said to be a repressible operon. This means that its transcription is usually on, but can be inhibited, repressed, when a specific small molecule, in this case tryptophan, binds allosterically to a regulatory protein. In contrast, the LAC operon is said to be an inducible operon, which means that it is usually off, but can be stimulated, induced, to be on when a specific small molecule called an inducer, in this case allolactose, interacts with a different regulatory protein and inactivates the repressor. The regulation of the LAC operon is similar to that of the TRP operon, but there is one important difference. While the TRP repressor is inactive by itself and requires tryptophan as a co-repressor in order to activate the repressor, the LAC repressor is active by itself and requires allolactose as an inducer to inactivate the repressor and turn the LAC operon on. The regulation of both the TRP and LAC operons are examples of negative gene regulation because the operons are switched off by the active form of their respective repressor protein. Gene regulation is said to be positive only when a regulatory protein interacts directly with the genome to switch transcription on. We see an example of this positive regulation with the bacteria E. coli. When glucose and lactose are both present in its environment, E. coli will preferentially choose glucose. However, when lactose is present and glucose is scarce, cyclic AMP, CAMP, accumulates and binds to a regulatory protein called cyclic AMP receptor protein, CRP. This regulatory protein is an activator, which is a protein that binds to DNA and stimulates transcription of a gene. When CAMP binds to this regulatory protein, CRP assumes its active shape and can attach to the LAC promoter. This attachment increases the affinity of RNA polymerase for the Lac promoter, thus increasing the rate of transcription of the Lac operon. When glucose levels increase again, the cAMP concentration falls, and CRP detaches from the Lac operon. With CRP in its inactive form, the affinity of RNA polymerase for the Lac promoter decreases and transcription returns to a normal rate. Since the attachment of CRP to the promoter directly stimulates gene expression, this mechanism is said to qualify as positive gene regulation. As you can see, the Lac operon is under dual control: negative control by the Lac repressor and positive control by CRP. Whether or not allolactose is bound to the Lac repressor determines whether or not transcription of the Lac operon's genes occurs at all. Whether or not cAMP is bound to CRP controls the rate of transcription if the operon is repressor-free. It helps to think of the Lac operon as having both an on-off switch and a volume control. I hope this video helped you understand the basic components and functions of the TRP and Lac operons, the mechanisms involved in positive and negative gene regulation, and the essential roles the TRP and Lac operons play in each. Thanks for watching!